Right, this is meiosis, genetics and inheritance. We will start by looking at genes and alleles. So, uh, oh no, it's green in color. Hang on, let's change it to white. Okay, we're good to begin. So let's start with a cell over here. We have a cell. This is, let's say this is a somatic cell. Okay, so what is a somatic cell? A somatic cell is any cell in your body other than gametes. So it's any cell in your body other than sperm cells and egg cells. And this is important because sperm cells and egg cells in your body are the only things that have 23 chromosomes. Whereas all the other cells in your entire body, your hair, your muscle, all that, they all have 46. So sperm and egg only have half. The reason being, so that when these two fuse together during fertilization, during intercourse to make a zygote, the first human cell, we get 46 again. Okay? It's to maintain that chromosomal number during reproduction. So that's a somatic cell. Let's say, for this example, this somatic cell over here is a muscle cell. Okay? This is a muscle cell. Muscle cell of for male. Okay, so now we're looking at a male's muscle cell. We know that the male has 46 chromosomes. All right, 46. So we know also in meiosis too that they line up in pairs. So if there's 46 chromosomes, there will be 23 pairs. Okay, I can't draw all 23 pairs, all 46. So there's the first pair, second pair, third pair, 23rd pair, 23 times 2, you get 46. Now, among these 46, we discussed that when fertilization occurs, sorry, we get 23 from our mom, 23 from our dad. So let me just draw here. Again, I can't draw all 23. Just give me a second. All right. There we go. Pretty bad, bro. But it's okay. So 23 from your dad, 23 from your mom. When they fuse, they form the first zygote. Okay, they form the first zygote. Sperm and egg fuse, they form the first zygote. This zygote becomes 2 cells, 4 cells, 8 cells. Mitosis. And this grows into a human being. Okay. Now, on each of these chromosomes, we have specific lengths of genetic material called genes. And different genes code for different proteins, and different proteins make different cells make different tissues make organs, and with these proteins, the basic building blocks, we make our physical characteristics. So let me draw that out over here. Let's say we have our 46 chromosomes over here. And from that, we take one small length of the chromosome. We use the genetic information here. Let's say the genetic information has a base sequence of that. We copy this, we make it into a messenger molecule called mRNA, and we send this over to an organelle or part of a cell called a ribosome. Now this is all extremely simplified. It's all extremely simplified. This process over here is called transcription. And this process over here is called translation. It's all extremely simplified, but this is just the gist of it. Let's say we take a small part of our genetic material, we copy its information, and we send it to a ribosome. So if you've learned about cells, you will learn that cells have different parts, like nucleus, like the Golgi, right? What other things can we name? We have mitochondria and all that stuff. So a ribosome is a part of the cell. And what does a ribosome do? A ribosome receives genetic information in the form of mRNA and uses it to make proteins. So that is what ribosome does. So let's say this gene over here was the instructions for hair color. Let's say red hair. So your chromosome sends out an mRNA instruction for red hair. It sends out this code for red hair. It goes to the ribosome. Ribosome reads it and makes the protein. What happens then is, if the ribosome makes enough proteins for red hair, these assemble to form red hair cells. Keep in mind that proteins are the basic building blocks. These proteins are not cells. Proteins are used to make parts of cells. So many proteins combine to form cells. Many cells become tissues. Tissues become organs, and organs become systems, etc. So let's say this was the gene for big biceps. Big biceps gene makes a copy of itself. It's read by the ribosome. Ribosome reads the gene and makes proteins. The proteins form muscle cells. It's a bit of a sketch here. It's fine. Muscle cells. Muscle cells aggregate in groups. Lots of them. Lots of them combine to form muscle tissue. And tissues form the organ being bicep. So that is the role the genetic information plays in making us. This is why DNA is called the building blocks of life, because it's the code for life. We read this code to make proteins, to make the building blocks, and the building blocks, the smallest units, build bigger units, and even bigger units, and then all these bigger units combine in systems and organs to form a fully functional and awesome human being. All right, that's great. So now we've discussed what a gene is. A gene has information for coding. Let me just remove all this here. So naturally, we know that all our chromosomes are different genes. So let's say there's one gene over here, one gene over here, one gene over here, one gene over here. And one thing to know as well is that in a chromosome, we know that this is a chromosome. This is a chromosome. We know that chromosomes are attached in the center by something called a centromere. And we know that each of these legs, each of these things, are called chromatids. What is very important to know is that chromatids are identical. So the left chromatid, the right chromatid are completely identical. That is why they are called sister chromatids. So let's say, let me just draw this better. Let's say we have a chromosome here. Let's say on this part of the chromosome, we have the allele for red eye color. So let's say this gene is for eye color, and let's say the allele here is red color. What's important to know is that this gene exists, this allele exists on the same location on the other chromosome as well. I mean, on the other sister chromatid. So chromatid combined to form chromosome. And since they are on roughly the same location, this estimated location is called a loci. Okay, so different alleles for the same gene because we're talking about the gene for eye color, exists in the same loci. So let's recap. We're talking about eye color. Eye color has genes. The genes can vary a bit. So different versions of the gene. So let's say I give A, T, T, C, C, A, T. Let's say this is the code for red eye color. I'm too lazy to color this red. Let's say A, T, G, C, A, T. We can see there's a one letter difference, a slight variation between these two codes for blue eye color. These different versions are called alleles. Okay? So when your mom and your dad release gametes and they fertilize, the gametes may carry different alleles. So let's say if your dad had the allele for red eye color and your mom had the allele for blue eye color. So let's say red eye color is the allele for has the symbol big R, and let's say blue eye color has a symbol small r. Okay, when they fuse, they form a pair over here. Big R, small r. Okay. So now there's a bit of a complication here. One version of the gene is telling us to make red. One version is telling us to make blue. What do we do? Do we make red eyes or do we make blue eyes? This is extremely complex in genetics and boils down to a lot of factors. But since this is a monohybrid cross, we can say that based on these two, one is dominant. Based on these two different versions of the gene, based on these two alleles, one is dominant, one is recessive. Excuse me. So let's say we choose big R to be dominant, so red eyes are dominant, blue eyes are recessive. So if we have a situation big R, small r, if you have a combination of two versions of the gene, two alleles from mom and dad, big R and small r, if we were to put them in a cell, in the end, we would have more proteins for red eyes being made. More proteins for red eyes being made than for blue eyes. So in the end, since there's much more red eye protein, a red eye is what manifests in the human being. So since this red eye allele is dominant, it overpowers the blue eye. So in the end, this organism ends up with red eyes itself. Okay? And this combination of the alleles is called genotype, the combination. So let's say you get the gene for blue eyes from your mom, blue eyes from your dad. What happens now? Do you get red eyes or blue eyes? The answer is you get blue eyes. Because even though these are recessive, even though these are weak gene, there will only be blue eye proteins being made. And hence, the only option is to make a blue eye. Similarly, if you receive the red eye allele from your mom and the red eye allele from your dad, only red eye proteins will be made. And as a result, a red eye is made. The problem only arises when we have clashing alleles, a dominant and a recessive. So when we have similar alleles, this is called homozygous. It's a single word. Okay, so these are both homozygous because they have the same allele. Mom and dad give the same allele. Over here, since mom and dad give different alleles, one's dominant, red eyes is dominant, blue eyes is recessive, this pair of alleles is called heterozygous. All right. So we have discussed that we get different variations of the genes called alleles, one from a mom, one from a dad, And depending on the pairing of the alleles, we get different characteristics. So if two dominants pair up, we get the dominant manifestation, we get the dominant phenotype. So we have red and red. The only protein we can make is for red eyes. So we get red eyes, the dominant characteristic. If we had a red allele and a blue allele, the red allele overpowers the blue allele. So much more red proteins being made than the blue allele. And hence we still get red eyes. All right. So that was look at genes and alleles. Let's see how all of this ties into reproduction. Awesome. Okay. Let's say, hang on, give me a second to clean up my sketch. Okay. Let's say we get 23 chromosomes from mom, 23 chromosomes from dad, and both the alleles for eye color, both the versions of the gene for eye color are dominant. Red, red. And this is the phenotype. What will be the color of this person's eyes? They'll be red color because only red proteins are being made. So this is a muscle cell of, or an eye cell. Let's say this is an eye cell since we're talking about eyes. Say this is an eye cell, okay? What's important to know is that eye cells are somatic cells, and so are these things in our body called germ cells. Germ cells are reproductive cells. These are the precursor reproductive cells, and germ cells undergo meiosis. Germ cells undergo meiosis. So you have meiosis 1, meiosis 2. Germ cells undergo meiosis to form gametes. So see this germ cell over here has now formed 4 sperm. Awesome. So let's say with 46 over here, we end up with 23 chromatids in each sperm. Okay, we don't need to worry about that much for now. Let's just look at this. So since the germ cell and the eye cell are similar, the germ cell would have exactly the same number of chromosomes, exactly the same number of the same genes and all that as the eye cell. The eye cell, somatic cell. So let's look what happens during meiosis. When meiosis happens, prophase 1, metaphase 1, anaphase 1, telophase 1, basically what happens is the homologous chromosomes are pulled to opposite sides. Okay, this is an oversimplification. There will be things like crossing over and independent assortment happening throughout meiosis, but we're just going to ignore that for now for the sake of simplification. So when the homologous chromosomes are split and pulled to opposite ends and cytokinesis occurs, we get this. The pairs have been split in half. So now each cell only has 23 chromosomes. Again, I can't draw all 23, but the point here is that these pairs have split up during mitosis 1. During mitosis 2, we learned that mitosis 2, the steps are very similar to, during meiosis 2, sorry. So during meiosis 2, meiosis 1, during meiosis 2, we learned that meiosis 2 has similar steps to mitosis. What happens is that the chromosomes line up and they are torn apart at the centromere, right? So after the mitosis 2, we are left with chromatids. Okay? Because during mitosis 2, these split. All right, this is a centriole. This is a simplified drawing. Awesome. So that's for one cell. The other cell had these chromosomes. All right, awesome. And now let's just keep track of our genes. We said that this is big R, this is big R. We said that each leg of, each half of the chromosome, each chromatid is identical. So if this is R as well. This is R, this is also R. So let's see down here. We have R, R, and we have R, R, because we got this, we got the same, we got big R from both our mom and dad. So in the end, when we split everything, each gamete has the allele for red eyes. So let's say these are all sperm. Okay. So these are all sperm. So all of these sperm that we've made from one germ cell have the allele for red eyes. Could there ever be sperm with a blue eye allele? No, because our parent cells, our somatic cells, never had the gene for blue eyes. So because when the sperm and egg combined over here to make the first cell, there was no blue eye gene. There's no blue eye gene here as well. So all our gametes will always contain the red eye, the big capital R gene. Okay. That is for your mom. Let's draw a similar diagram for dad. This is for dad. Let's draw a similar diagram for your mom. But let's change it up a bit. Let's say this is your mom. Okay, your mom has 46 chromosomes. 23 from her mom, 23 from her dad. And let's say your mom has blue eyes. Let's say your mom has blue eyes. So we're saying the dad here, dad has red eyes. Okay, let me just draw that over here. The dad has red eyes because his genotype is RR. Capital R, sorry. And let's say the mom, okay, has, let's say the mom has blue eyes. She would have blue eyes because both alleles are recessive. So the proteins made are blue. So let's say smaller over here, okay? So let's label all the genes systematically. We said the genes from both mom and dad appear in the same loci. So we're saying that both sperm and egg that made the mom had the blue eye gene now. So there's no red eye gene involved here. Let's say RR. Here also is R and R. And we make our gametes. We can say that 1 second. We can say that all of these have one gene, one allele, sorry, the only allele possible for the blue-eyed mom, which is smaller. So let me just label that over here. Oh no. Smaller, smaller, smaller. Whoops, it's not supposed to be an R there. Smaller and smaller. Okay, perfect. So now we have gametes from the mom and from the dad. All right, so let's look at that again. We had a germ cell from my dad, a germ cell from my mom. We discussed that the dad had the genotype big R, big R. So the dad has red eyes. And we said that the mom has genotype small r, small r. So by default, because these are both recessive, she has blue eyes. These are called genotype. These are called phenotype. So genotype is the gene configuration of the alleles. Phenotype is the trait that results from the production of the proteins and all that. Let's see what happens when you fertilize. Okay, we know that in reproduction, one lucky sperm meets one egg. Only one egg is released into the fallopian tube. This is extra biology. The other three disintegrate. So now, one sperm and one egg fuse. As a result, when they fuse, this is how fertilization works by nature. All the chromatids all the chromatids end up in the same cell. Okay, let's look at that. I'm only drawing 4, but there's actually 23 in each chromosome. 23 in each gamete. So all of the chromatids, this is a chromatid, all the chromatids join together, fuse. They unite in this cell. They replicate their genetic information. And we have a human zygote with 46 chromosomes again. And as we said just now, let's see the two gametes that we're fusing in fertilization. One of them has the gene for red eyes, one of them has the gene for blue eyes. So the dad is giving the gene for red eyes. He's giving the gene for big R, the allele for big R. The dad is giving the allele for small r, for blue eyes. Okay? As a result, will this offspring have blue eyes or red eyes? The answer is, since this is heterozygous, the red eye gene is dominant. More red proteins are being made than blue proteins. As a result, the phenotype is red. So let's take a minute to recap what we've just looked at. We have discussed what somatic cells are. Somatic cells are normal cells in our body. We've discussed that genes, the information from genes is used to code for proteins. The mRNA is sent to ribosomes, and then proteins are made. Proteins are the basic building blocks of life. These proteins, along with other biological molecules, are used to assemble cells. Cells make tissues, tissues make organs, etc. We have discussed that from a mom and dad, we can get different variations of the gene called alleles. So from our dad, we could get bigger. Our mom, we can get smaller. And depending on whichever gene is dominant or recessive, there's a bit of a competition here to decide whose allele gets to influence the characteristic of the offspring, whose allele decides the phenotype. So it's always the dominant allele that wins. And in this example, we discussed that big R, the allele for red eyes, is dominant. So therefore, the offspring has a phenotype of red eyes. And we have drawn a complex diagram over here and slowly unraveled and observed the transition of these genes. So from the dad cell, the big R genes in both the mom and in both his mom and dad chromosomes. Somehow we saw how those form gametes. Mom cell form the gametes as well. And the mom and dad are giving clashing alleles. In the end, it's the dominant allele that wins. So based on this genotype, the phenotype is red eyes.